Today we're going to talk about explicit or conscious memory in animals. This is the second part in a two-part series talking about the mechanism of learning. The first part discussed implicit memory, which is unconscious memory, where we went into great detail of the chemistry and simple organisms such as Aplysia sea slugs. This series is also based off the same review written by Eric Kandel and his colleagues. In this first section, we're going to focus on what we have learned from mice about explicit memory. The building blocks that were explored in Aplysia and Drosophila are going to be further developed and elaborated on in the mammalian brain. In the study of mammalian brains, there is much difficulty in associating a certain stimuli with a brain region because there is so much stimulation that is happening in a mammal at one given time. Determining the input and the output is not easy. We either have to look at the gross level of the mammalian brain or a single cell level. Remember, from the last section, information is encoded in the strength and connectivity of neurons. In this episode, we're talking about facts and events that are remembered. And I've learned that these bits of information are stored in the hippocampus. The hippocampus was found to be important in the study of amnesia patients. So when there was a lesion or damaged part of the hippocampus, these patients had trouble with memory. Space happens to be a readily analyzed type of information in mice. There are cells in the hippocampus that exhibit a spatial specific firing in freely behaving rats that are monitored using electrophysiological recording. Certain so-called place cells are activated when the animal is in a certain space. Unlike sensory and motor cortex, place cells are not organized in the hippocampus. In mouse, the space that is perceived is not mirrored or replicated in the hippocampus. The space around is encoded in different cells throughout the hippocampus. However, if a certain location is focused on repetitively, those same cells will fire. Attention is required for encoding and retrieval of locations in place cells. The stability of firing of the place cells and the completion of spatial tasks in mice correlates with the amount of attention. This makes sense to me. Have you ever tried navigating without a GPS, realizing that you have greater familiarity with those streets than with a GPS? The specificity of place cells takes some time to develop, so the network can be modified as the animal perceives the environment. Grid cells in the enterohynal cortex actually communicate with the place cells and convey to the hippocampus about space. Research has shown that grid cells are maintained while the hippocampus cells adapt. So it could be a more hardwired understanding of space. Monkeys have been shown to have a decrease in grid cell firing for repeated stimuli, so there is a link between grid cells and learning. Another study showed that objects that were placed in a specific location activated specific neurons in grid cells when the object was put in the same place as a previous trial. So this is a readout of previous experience. Other cells in the same enterohynal cortex would respond to the object's presence. So this separates object cells and object trace cells. In order to develop long-term memory, researchers looked at pulsed studies. They would activate neurons at different frequencies, and they discovered that long-term potentiation or long-term memory was formed with continual stimulation at a variety of frequencies. They found that 100 Hertz stimulation resulted in long-term potentiation based on a NMDA glutamate receptor activation. These receptors are both ligand-gated and voltage-gated. When active, these receptors bring calcium to the postsynaptic cell, and that's needed for long-term potentiation. Calcium begins a signaling pathway with various kinases, including PKA and MAPK, that we found were essential for long-term potentiation for implicit memories in the last episode. These pathways are also modulated by dopamine, which means that plasticity can be altered by attention and motivational state. Early in LTP, messengers increase AMPA receptors, strengthening the synapses as well. So for long-term potentiation, we discussed that protein synthesis is required for implicit memory. In explicit memory, the same applies. In PKA, MAPK, and CREB-B, the same players are required to express genes for long-term potentiation. Similar long-term potentiation can be made from different patterns of stimulation, not just a 100 Hz frequency, with various contributions from NMDA receptors. Physiological patterns of stimulation were somewhat induced by pairing a presynaptic stimulus with a postsynaptic stimulus, separated by about a millisecond, which is true for natural firing of neurons. This is called spike timing dependent long-term potentiation, or STDP. If the order of stimulation is reversed, the connection is weakened, and this is dependent on NMDA. It's the reverse of long-term potentiation, called long-term depression. Researchers were able to eliminate the NMDA receptors from certain cells in mice using a Cre recombinase. They had flanked the NMDA gene with LOXP sites and were able to delete that gene through recombination. When done in the dentate gyrus of the hippocampus, there was a loss of long-term potentiation. Mice did not learn to fear one of two chambers that they were shocked in, like the control mice were. Plasticity changes in response to other transmitter systems as well, and by past stimulation history of a particular synapse. And we've talked about consolidation, converting short-term memory into long-term memory. But it's also true that memory can be reinforced by recollection, and this is called reconsolidation. There's actually much less known about reconsolidation than consolidation. Now I'm going to discuss memory in humans. fMRI is a method that has been used in the last few decades. It detects blood oxygenation level-dependent signals, which is a fancy way of saying it detects blood flow. And blood flow in the brain correlates to neural activation. There's a subsequent memory paradigm, which is employed in experiments where a brain is monitored during a learning session, and then it's monitored again at a later time when the information is being remembered. This type of experiment allows for the learning of regions in the brain where the information is stored, and also the regions that are used for encoding and retrieving the information. Researchers found that the coding and retrieval consistently happens in the medial temporal lobe, which includes the hippocampus, spaces in the prefrontal cortex, and the posterior parietal cortex. And this was predicted in amnesia studies and also animal models. It has been known that our brains reorganize information in wide regions of the brain for long-term memory that lasts months and years. The standard consolidation theory is that the hippocampus memory decays as the neocortical reorganization occurs. The roles of the brain areas during reorganization is up for debate, though the mechanisms tend to involve the medial temporal lobe, especially the hippocampus. So when the hippocampus was silenced using a halorhodopsin-based method, which is where cells are producing this receptor that's sensitive to light in certain cells, and here the whole hippocampus was designed to activate with light, and when the hippocampus was activated, silencing its activity for much of the time between the fear conditioning and the recall, there wasn't really effect on the memory of the rodents, unlike shorter silencing episodes. And this suggests that there's some compensatory mechanism that allows for remembering without the hippocampus. This reminds me of an episode in the PBS series The Brain by David Eagleman, where a child undergoes a hemispherectomy, which is a complete removal of half of the brain, an entire hemisphere. And this is usually performed in an epileptic patient who is drug resistant. Incredibly, the patient recovered with full cognitive ability because of neuroplasticity. Consolidation is hugely affected by sleep or lack of sleep. And this agrees with Matthew Walker's book, Why We Sleep, that I read a few years ago. There was a study where patients associated a sound with an object, and the researchers found that the sound only reinforced the association when it was played while they were sleeping. Does this mean that you can study more effectively by replaying a recording of yourself reciting one of your course chapters? Seems possible, although I think it would have to be proven in another study. The brain can retrieve elaborate information in less than a second, but we don't know much about this process. The fMRI cannot resolve this because its resolution is on the second timescale. We expect both sequential and parallel processes to be involved. We also know that the medial temporal lobe is necessary and that it interacts with the prefrontal cortex. The parietal cortex is probably involved in attention-driven retrieval. Using a different method called electrocorticography, patients being monitored for seizures in different areas of their brain were found to have greater connectivity between certain regions. This was measured by phase synchronization. Certain regions had better synchronization through time, meaning the pulses of a signal through different regions were similar, associating them together. It makes sense to look at frequency of signal in epileptic patients because seizures are actually a result of uncontrolled pulses of activity. In these patients, they found that a more accurate retrieval was associated with better connectivity of these different regions. The mechanism of learning and remembering in humans is incredibly complex and mysterious. There were some important questions that were asked in Kandel's review, including, is memory stored in population rate codes or simply the connectivity and strength of synapses, or is there also a spatiotemporal code where frequency of pulses encodes information? We know that there are frequencies that result in long-term potentiation, but do frequencies also encode for specific memories? Also, is the learned information spread across large areas, or is it concentrated in particular areas? Unlike what we've seen in the hippocampal representation of space. That is all I have for this series on memory. I hope I didn't confuse you too much, or at least I hope you have become more curious about this topic that I find so interesting. Have a good one.